This video covers Chapter 17 notes in your book, which is protein synthesis. We will look at transcription and RNA processing and translation. Gene expression is the process by which DNA directs protein synthesis, and it includes both transcription and translation. So here in figure 17.4 is a basic overview of transcription and translation. And transcription is the process of using DNA to make RNA, specifically messenger RNA or mRNA, which is one of the three types of RNA. In eukaryotic cells, that process occurs in the nucleus, and in prokaryotic cells, it occurs in the cytosol. After transcription, eukaryotic mRNAs are modified through RNA processing before translation begins. During translation, ribosomes use the mRNA to make a polypeptide chain, which will be adjusted and folded and modified or whatever before it is a functional protein. Primary transcription is the initial mRNA transcript that's made from a gene before processing. And all of this is part of the central dogma of biology that says that if we have DNA, it can be used to make more DNA during replication, or it can be used to make RNA during transcription, and then that RNA can be used to make a protein during translation. We'll start by looking at the genetic code. So our question here is, how are the instructions for assembling amino acids into proteins encoded into DNA? There are only 20 amino acids, but 4 nucleotide bases in DNA. So then the question becomes, how many nucleotides correspond to an amino acid? And the answer comes is that mRNA is read in groups of 3 called a codon. One of the strands of DNA will be the template strand that provides the template for the order of the mRNA nucleotides. Whichever strand is the template is always going to be the template strand for that gene, and it's determined by the orientation of the enzyme that transcribes the gene. Those mRNA codons will be read from 5' to 3', and each codon gives us 1 amino acid. The non-template strand is called the coding strand because it will have the same sequence as the mRNA strand, except there will be uracil instead of thymine. And of course, it's RNA nucleotides over DNA nucleotides, but you get the idea there. And so this is the codon chart that lets us know which codon corresponds to what amino acid. All 64 codons were deciphered in the early 1960s. 61 of them code for an amino acid, and 3 are stop codons that signal the end of translation. There are some repeats, meaning more than one codon gives the same amino acid, and the codons have to be read in the correct reading frame or in the correct group of 3 to make the correct polypeptide. And you'll notice that there is only one start codon that signals start translation here, and it is methionine. So all the proteins that are made, that will be the very first amino acid. The genetic code is nearly universal. It is shared by bacteria and complex animals. And what that means is that genes can be transcribed and translated after being transferred from one species to another. So there's 2 examples here. In figure 17.7a, you can see the tobacco plant is expressing a firefly gene, specifically the gene that makes it glow. And in 17.7b, you can see a pig expressing the jellyfish glow gene. So because that can happen, we know there's some universal connections there. Now we'll move on to the process of transcription. An enzyme, specifically RNA polymerase, separates the 2 DNA strands and joins RNA nucleotides that are complementary to the DNA template strand. It is going to assemble RNA nucleotides in the 5' to 3' direction. RNA polymerase is able to start the chain from scratch, so it's not going to need a primer like is needed for DNA replication. And if you'll remember, that primer in DNA replication is a little bit of RNA. Specific DNA nucleotide sequences mark where the beginning and end of transcription of a gene are. And RNA polymerase will attach and begin transcription at the promoter region. And we'll look at termination here in a minute because it's going to be different between prokaryotes and eukaryotes. The direction of transcription can be referred to as upstream or downstream, depending on which strand's being used and which direction the process is going. There are 3 stages of transcription: initiation, elongation, and termination. And you can see in figure 17.8, it's kind of a general picture of all 3 of those processes that we will now look at in detail. Starting with initiation. The promoter region of a gene includes the start point of transcription and extends several nucleotide pairs upstream. RNA polymerase binds in a precise location and orientation on the promoter, and that's going to determine where transcription starts and which strand is the template. In prokaryotes, RNA polymerase recognizes and binds to the promoter, but as always in eukaryotes, it's not that simple. Several things have to be in place before transcription can start. First, there have to be transcription factors to mediate the binding of RNA polymerase and initiation, and that whole complex is called the transcription initiation complex. The ta-ta box is a crucial promoter DNA sequence, and all of that has got to be there and in the right place before transcription begins. So here in figure 17.9, this is eukaryotic transcription. There's the ta-ta box, that's part of the promoter region, and then a little bit downstream is going to be the start point. Then the transcription factors are there, ready to go, and then RNA polymerase can bind and start transcription. Next is elongation. RNA polymerase untwists the double helix as it goes, which exposes 10 to 20 DNA nucleotides at a time. RNA polymerase will add nucleotides to the 3' end of the growing mRNA transcript, and the new RNA or mRNA molecule will peel away and the double helix can reform. The rate of transcription is about 40 nucleotides per second in eukaryotes, and genes can be transcribed simultaneously by several molecules of RNA polymerase. And so like one starts and then another and then another and another. And that's important to, 1, increase the number of mRNAs and therefore make the protein in larger quantities, because 9 times out of 10, just making that one protein is more than 9 times out of 10. But cells are going to need more than one protein to be able to accomplish the task. And so that helps speed that process up and make sure there are appropriate quantities of those proteins in the cell. And there is a diagram of elongation. Last is termination, and we'll start with prokaryotes and move on to eukaryotes. In prokaryotes, transcription continues through a terminator sequence in DNA, and that transcribed terminator acts as the termination signal, and so RNA polymerase is going to detach from the DNA and release the transcript. In eukaryotes, RNA polymerase transcribes the polyadenylation signal sequence, which specifies a polyadenylation signal in the pre-mRNA. And it's pre-mRNA because we know it needs to go through some processing before translation. Once that signal is reached, it is immediately bound by certain proteins in the nucleus, and then 1 to 35 nucleotides downstream of that sequence, the protein cuts the RNA transcript, and then we're ready for RNA processing. The 5' end of the mRNA transcript is going to get a cap, which is modified from a guanine nucleotide, and it's added after the first 20 to 40 nucleotides during transcription. It can also be called a GTP cap. The other end, the 3' end, gets a poly A tail, and so there is an enzyme that adds 50 to 250 more adenine nucleotides. And both of those are important for several reasons. 1, they facilitate the export of mRNA from the nucleus so that it can leave. 2, it protects the mRNA from degradation as it's leaving the nucleus and making its way to a ribosome. And third, it does help the ribosome attach to the 5' end. That cap does. So these modifications will have to be done before translation. In eukaryotes, there's also going to be a little bit of splicing that happens. RNA splicing is when portions of the RNA are removed and the remaining portions are reconnected. The non-coding segments are called introns, and the coding segments are called exons. And the removal of introns is done by a spliceosome. Ribozymes are catalytic RNA molecules that function as enzymes and can splice RNA. So what's going to happen if you look at figure 17.12 is those intron regions, or like the lighter red color, are going to be removed, and then what's left is what codes for the actual protein. Introns are important for lots of reasons. One, there are possible adaptive benefits. Two, some introns contain sequences that regulate gene expression and many gene products, even if they're not part of the overall protein. That also means that a single gene can encode for more than one kind of polypeptide. So it could be that depending on the way it's spliced, maybe there's like 3 different polypeptides that can be made from one pre-mRNA. Many genes will give rise to 2 or more different polypeptides depending on how it's sliced, and that's what we call alternative RNA splicing. It's alternative because depending on which way you splice, you're going to get a slightly different polypeptide. Next, we'll move on to translation, which is using that mRNA transcript after it's been processed in eukaryotes, or after it's been transcribed in prokaryotes, is going to be used to make a polypeptide. First, we'll look at the molecular components of translation. The cell translates mRNA message into protein with the help of transfer RNA or tRNA. tRNAs transfer amino acids to the growing polypeptide in a ribosome, which is why it's called tRNA. Every tRNA carries an amino acid on one end, has an anti-codon on the other end that will be complementary to the mRNA codon. For example, if the mRNA codon is AUG, the tRNA's anti-codon will be UAC. It is a single RNA strand that's about 80 nucleotides long that will be twisted and folded into a 3D molecule. It's about L-shaped with 5' and 3' ends, and the 3' end is the attachment site for the amino acid. And so in figure 17.16, we see like 3 different visual representations of the tRNA, but going forward, we're going to use C because it's easier to look at. And so that's what we're going to do. But if you like the other 2, good for you. Accurate translation requires the correct match between tRNA and amino acid and the correct match between anti-codon and codon. So tRNA has to have the right thing and they have to match up correctly. There is some flexible pairing at the 3rd base called wobble that will allow some tRNAs to bind to more than one codon. Ribosomes facilitate protein synthesis. Each ribosome has 2 ribosomal subunits that are made up of protein and ribosomal RNA, or rRNA. The size of these subunits are slightly different in prokaryotes versus eukaryotes, but they both have a large one and a small one. Ribosomes have 3 binding sites for tRNAs. The P site holds the tRNA that carries the growing polypeptide chain. The A site holds the tRNA that carries the next amino acid to be added. And the E site is the exit site where discharged tRNAs can leave the ribosome, which you can see in figure 17.18. So the exit site, the P site where it has the growing, and then it has the A site, which is what amino acid will be next. And those amino acids will be connected via peptide bonds. This is why it's called a polypeptide chain. There are 3 stages of translation: initiation, elongation, and termination. All of them require other protein factors for the process to occur, and some steps will require a little bit of energy. We'll start with initiation. The start codon, which is AUG, which gives us the amino acid methionine, signals the start of translation. The small ribosomal subunit binds with mRNA and the initiator tRNA. The small subunit moves along the mRNA until it reaches the start codon. So the start codon is not going to be the very first thing that is there. It's going to be a little bit downstream. Initiation factors bring in the large subunit, which completes the translation initiation complex, which you can see to the right in figure 17.19. With elongation, amino acids are added to the C terminus of the growing chain. It requires codon recognition, peptide bond formation, and translocation. Both codon recognition and translocation require an energy expenditure, and as always, translation is going to go in the 5' to 3' direction. Elongation is going to continue until a stop codon is reached at the A site of the ribosome. And when that happens, the A site accepts a protein called release factor, which is going to add a water molecule instead of an amino acid to the end. And that reaction is going to release the polypeptide to go do whatever it has coming up next. Translation is not sufficient enough to make a functional protein. The polypeptide chains will be modified after translation or targeted to specific sites in the cell. So starting with modification, the polypeptide chain will begin to coil and fold as it's being translated to give those secondary and tertiary structures that we know proteins have. But modifications may be required before that protein can effectively do its job. With targeting specific sites in the cell, free ribosomes synthesize proteins that function in the cytosol. Okay, so they're just the ones that are floating around. The bound ribosomes, which are bound to the endoplasmic reticulum, synthesize proteins of the endomembrane system and proteins that need to be secreted from the cell. Remember, it has to go through that whole endomembrane system of like ER, Golgi, etc., before they are ready to do their job. And here in figure 17.25, you can see an overall process of eukaryotic protein synthesis that includes transcription, RNA processing, and translation. Last, we're going to look at some mutations that can happen. A mutation is a change in the genetic information of a cell. A point mutation is a change in one nucleotide pair of a gene. If that mutation has an adverse effect on the phenotype, we call that a genetic disorder or a hereditary disease. I want to add in a note here that not all mutations are bad. Some of them lead to new traits in a population, and we'll look at that more when we get to Unit 7, Natural Selection. There are two categories of point mutations. First is a substitution, where one nucleotide pair is replaced with another. And those can be silent, missense, or nonsense. If the mutation is silent, that change still gives the same amino acid, so the polypeptide sequence is going to be the same. With a missense, the mutation codes for one different amino acid, and a nonsense happens when the amino acid is changed to a stop. With insertions and deletions, which are the second category, an insertion is an addition of a nucleotide, deletion is a loss, and those are going to alter the reading frame. And what I mean by that is we know mRNA is read in groups of 3 called a codon. So if we add one in or take one away, those groups of 3 are going to be adjusted and different from that point on. Spontaneous mutations can occur during DNA replication, recombination, or repair. Mutagens are physical or chemical agents that can cause mutations, which we know does happen, particularly since,